So my name is Carl Latham, and I'm head of commercial operations at ONTOFORCE. And our talk today will be on creating a sustainable and fair ecosystem supporting new insights in life sciences. And our proposal to achieve this is the integration of Irods with ONTOFORCE's knowledge platform, Discover, to offer a sustainable infrastructure to store and search for data, knowledge, and insights. Before I go deeper into our proposal, I'd just like to briefly introduce ONTOFORCE to you today. So ONTOFORCE, our mission is to help our customers to unlock hidden insights from data by breaking down boundaries between silos to make data discoverable, accessible, and usable. We ultimately contribute to increasing the quality of patient life. The company was actually founded in 2011 by Professor Hans Constant. And prior to founding ONTOFORCE, Hans was a data scientist at Eli Lilly for 10 years. And the original idea behind the Discover platform was to bring together disparate data on disease indications for healthcare professionals. In 2016, the company pivoted to working with pharmaceutical companies, and today our customers range from top 10 pharmaceutical companies to mid to small biotech companies, research institutes, and academia. We're based here in Ghent, but you can probably tell from my accent that I'm not from East Flanders, I'm from London. So we are a growing international company, and we were recently featured in Deloitte's Fast 50 technology companies in Belgium. Now, I don't think it will surprise many people here today when I say that data in the life sciences industry is growing exponentially, both in complexity and volume. And as a result, one of the biggest challenges is maintaining a view of all available data within an organization. And furthermore, there's a real desire to democratize access to data to the end user and the researcher to be able to find insights from this ever-increasing volume of data. But how does that desire to access more data and insights, how is that a reality today? Well, on a given day, if you are a clinician, a competitive intelligence professional, or a bioinformatician, you likely have a number of questions that you would like to find the answer for. So it could be a clinical feasibility question, as an example. You might want to understand the number of pancreatic cancer trials in phase 2 that are currently recruiting for patients in Germany. If you're a competitive intelligence professional, you might be interested in understanding how many companies are working on the target APOL1. Or if you're a bioinformatician, you might be interested in obtaining sequencing data from all oncology trials that have reported cardiovascular adverse events. But the feedback that we get from researchers is that really getting the answers to these specific questions is not an easy thing for them to do. And typically, the journey starts for the researchers by going into something that we refer to as the data swamp to get the answer to that question. And here we see two key challenges for the end user. So the first is that data is siloed. To retrieve an answer to that question, you might have to go to different knowledge platforms to get an answer to your question. And typically, data is not harmonized across all of those different data silos. So as an example, if you're searching in one data platform for the active substance dasatinib, you might then need to go into another platform to search for the marketed drug Sprycel, and you would need to know that there's a link between that active substance and that drug. So as well as having to explore those different data silos, you also need to check that you've captured all of the correct synonyms for the drug or the active substance that you're looking for. And the end result, according to Gartner, is that researchers are spending on average a day a week, 20% of their time searching and looking for information. At the same time, there's an increasing interest to explore combining multiple data sets and integrating those sources. And to achieve that, we believe at ONTOFORCE that using fair data is essential, because without uniform protocols throughout the data cycle related to things like standardization, metadata provision, and quality assessments, data originating from different sources is really difficult to compare. The FAIR principles of making data findable, accessible, interoperable, and reusable have been taken up by many data-driven communities since its publication in 2016, with grant and paper submissions often having a requirement now to explain how data will be made fair. And with FAIR data implemented and integrated, research queries can be answered more rapidly. Researchers can place more focus on data interpretation rather than collection or recreation, thereby driving greater research efficiency. And valuable data sets can also be created in shorter timeframes, leading to innovation faster and less time spent in data handling. And at ONTOFORCE, we are a proponent of the FAIR data principle. So within our Discover platform, we ingest 140 different public data sources into the Discover platform. So sources like clinicaltrials.gov, UGM2022, WHO, Open Targets, and Kemble. We also include ontologies like ICD-10, ICD-9, and MESH to ensure that data is fair and harmonized across those 150 public data sources. Now I'd like to introduce to you our Discover platform and our proposal to achieve the integration with Irods to offer a sustainable infrastructure to store and search for knowledge, data, and insights. The Discover platform enables data-driven decisions and accelerates research by unlocking siloed data. So the platform integrates and harmonizes data sets across internal data, public data, and third-party data sources into an integrated knowledge graph. And there is a consistent and easy-to-use interface that democratizes access to data through self-service knowledge discovery, allowing each scientist to access and explore data and to generate insights. Discover therefore helps you to do your research in one place, to answer complex questions and solve problems. And I'll actually be going into a short live demo of the Discover platform after the presentation. On this next slide, I'll explain the integration of Irods and Discover. So as data and metadata is captured in various source systems centralized in Irods, data can eventually be processed with an entity extraction service to further enrich metadata. You can also link metadata sets originating from different systems, specifically data sets stored within your Irods environment. And they're made findable in Discover by transforming the metadata into RDF triples and importing those as data sources into the Discover platform. Once the relevant data and metadata is loaded on the Discover platform, the data is then integrated and harmonized using the various ontologies and reference data sets that I mentioned before. A key point that I'd like to mention here is that the Discover platform actually enables users to simultaneously search through public and in-house linked data sets without having the expertise of writing Sparkle queries. And researchers can now very easily identify, search, and export available data on Discover and redirect their request to Irods to access the original data. And what we'd like to propose is that end users will use Discover as a preferred way of searching through Irods metadata while finding related metadata from public sources as well. So we believe that the integration of Discover and Irods platform provides a self-service access for research and a sustainable data ecosystem. And we believe that such an integration has broad applicability supporting research and development within the life sciences industry. So I mentioned earlier that our Discover platform is built using an underlying knowledge graph. And at this point, sometimes we hear feedback from the bench scientists that they don't really care about knowledge graphs. It's very complicated and ultimately they're not a data scientist. And I really think that this is where the power of our Discover platform comes into play. So whilst the knowledge graph enables that exploratory search between different data sets, the user is shielded from underlying complexity by our flexible, intuitive user interface. And the user interface can be customized according to the user's preference to ensure that information retrieval and exploration is simple and effective. And most importantly, it doesn't cause frustration for the researcher or the end user. Furthermore, the Discover platform is also quite flexible in enabling you to build plugin architecture. So we've recently collaborated with Chemaxon. So if you're a medicinal chemist and you would like to do a chemical structure search in the Discover platform, there's now an integrated plugin that enables you to search that way. One question that we get from prospective partners or customers when it comes to fair adoption and knowledge solutions is where do we get started? How much does this cost? What are the resources involved? And how do we demonstrate a return on investment to our stakeholders and sponsors? And I think the feedback that we've had is that trying to approach fair data adoption at the enterprise level comes with many pitfalls, because as I mentioned, the cost, the resources, getting executive buy-in, and also the change management needs to take place. And the feedback that we've received from companies like Merck, AstraZeneca, Bristol Myers Squibb is to mitigate these pitfalls is to start with fair adoption at the use case level. So by building a fair knowledge solution that supports a specific use case, for example, target identification, you can take your first step on a pilot project which requires less resources and lower initial cost to demonstrate the value of fair adoption. And companies that we work with have achieved success by really understanding the specific needs and challenges of the bench scientist, and then measuring the impact of the adoption of a fair solution use case like this before and after implementation, and also measuring the research outcomes. I'll briefly introduce one such pilot with Princess Maxima, who are a children's oncology research institute in the Netherlands. So the pilot was set up as a prototype of a fair data station using our Discover knowledge platform. We started with 14 data sets originating from both clinical and preclinical research. So metadata processing pipelines were built that uniformized the various data types, and during the processing, all data sets were actually translated into RDF format, which facilitates the use of a machine-readable semantic data model. This then in turn allows the implementation of inferred relationships between commonly used data concepts, leading to a tightly interconnected network of data sets. The resulting integrating data is then visualized on the dashboard that you see here, and that allow for cross-data set exploration of those sources that are interconnected in the underlying knowledge graph. And you can also see that the dashboards were configured to the Princess Maxima user specifications to support easy data retrieval for their clinicians and their researchers. I'm now going to jump into a live demonstration of the Discover platform. So this is not without, let's hope that the Wi-Fi works and this doesn't go down while I'm going through this. So this is the landing page of the Discover platform. Oh, sorry, is that not? Yeah. Just. Okay, no problem. So the first thing I'd like to show you is, as I mentioned before, we have the 140 public data sets that we pre-ingest into the Discover platform. So you'll see sources like PubMed and NCBI. You can also see a visual representation of the knowledge graph and how the different canonical data types connect. So you can start with a gene target information and then see how that links to transcription pathways, publications or company information. So one of the searches that I wanted to look at today was a target that I mentioned earlier, which was APOL1. So you should be able to see at the bottom here that we're picking up 824 publications for APOL1 within the Discover platform. But using the ontologies that I mentioned earlier, yep. You'll see that we are tracking using the ICD-10, ICD-9, and MESH ontologies, the different synonyms that are associated with APOL1. So by including to cover, I can see that the actual number of publications has increased only a little bit, but also I think the important point there is to mention that for the researcher, they're not potentially missing any valuable data that they'd like to obtain on that target. Once I find my exact target match that you can see here, APOL1, I've highlighted all of the contributing data sources that make up this specific gene. So some of those that I've mentioned before, like open targets. And you can also see here how you're able to then link to the different canonical data types using the knowledge graph. An interesting point here is that you can also retain traceability of the original provenance of the data source by highlighting over this legend here to see where the original source was located. As a researcher, I might then be interested in understanding, okay, with this target, what diseases is this target associated with? So one that I looked at earlier was chronic kidney disease. And then I might also be interested in looking at the clinical studies that are associated with CKD. So you can see here that we've been able to retrieve over 6,000 clinical trial results. What I would then do is refine that to APOL1, and I can see very quickly that from 6,000 trials, I've been able to get very quickly to 6 results. And I can see that there are 4 recruiting trials with APOL1. If I'm then interested in investigating those trials in more detail, I can again see the source information that populates those clinical trials. And then I might want to explore who the organizations are involved, who the principal investigators are there any associated publications, and also gives me the ability to link out some more kind of scientific information. So what you're seeing here is just the public version of our Discover platform where we're integrating the public data. What I'd just like to briefly touch on to wrap up the presentation is the connection with Irods. So I'm just going to switch back to my slide deck. Okay, great. So if we want to add Irods as a source, we can connect the Irods REST API and then run ETL scripts to convert into RDF, which is in turn converted into the Discover triple store. And the end user can then use Discover on metadata extracted from Irods. So using an URI, you would see Irods as a source matched against the gene that we looked at before if there was a match, so APOL1. And then you can link out to a landing page on the Irods browser, as you can see here in this example, to retrieve the data. So in conclusion, our proposal today was to achieve the integration of Irods to offer a sustainable infrastructure to store and search for knowledge, data, and insights. We see the key benefits for pharma, biotech research institutes, and academia with the integration being that data can be reused and reprocessed in a fair way. Results being instantaneous as opposed to having to wait days or weeks to get an answer to your question. There are limited costs and resources to maintain this ecosystem. And with Irods, the clear benefit is the reduced data duplication and storage costs. And finally, the ability to easily link and harmonize public, private, and public data sources and third-party into one simple application. So thank you for your interest in our proposal today. Should you have any questions, I'd be happy to speak during the lunch session today. But I've also put my contact details up on the screen, so if you want to reach out to discuss this particular proposal or any of our fair data use cases, I'd be really happy to hear from anyone. That's it.
All right.